Welcome to Knowledge Without Bar. Today we'll discuss on a general introduction of viruses. So before we go into further detail, please subscribe our channel. It will gives us motivation to make more such videos. So let's start today's session. So what does uh, we all know that uh, viruses are the simplest type of life forms present on the earth. Usually, viruses uh, are called the viral particles as they are not typical cellular structures, and these specialized particle-like bodies have some peculiar characteristic features. For example, they are showing living properties when they are inside a host cell, particular host cell, but they do not have any replicative or living characteristic feature when they are present outside the cell. And the study of the viruses are called the virology. And the foundation of the subject virology was started date back to 1892 when a Russian pathologist Dmitry Ivanovsky uh, have started the investigation on a plant pathogen. This is a tobacco leaf infected with a virus. That time it was not known, and the disease, based on the symptoms, the mosaic symptoms, the disease is called the mosaic uh, disease, tobacco mosaic disease. So Ivanovsky was started to investigate what is the causal organism for this tobacco mosaic disease. He was crushed the leaves with the symptom and tried to investigate or see under the ultramicroscopic filter, but he failed to detect any uh, particular structure or any viral or bacterial structures. Because this is why Ivanovsky was unable to see the viral particle, because viruses, we all know the viruses are very small or teeny structure. It is very difficult to identify under a normal uh, complex microscope or compound microscope. So, for that reason, Ivanovsky couldn't find any structural body. But when this isolated um, juice, or what is called the mm, uh, crushed material from this disease sample, when inoculated to another healthy leaf, the healthy leaf shows the similar type of symptoms. That means the there is something, Ivanovsky stated that there is something present in this disease leaf which is capable to spread the disease on a healthy one. So that laid the path for studying the viruses, and later on different viral particles have discovered, and it was also discovered that this tobacco mosaic disease was actually caused by the tobacco mosaic virus. Is named upon the disease called, and that's why Ivanovsky is designated as a father of virology. So what are virus or virion? Viruses are genetic elements that cannot replicate independently of a living cell, uh, and the cell on which they replicate are called a host cell. However, viruses do possess their own genetic information and thus independent of the host cell's genome. Viruses rely on the host cell for energy, metabolic intermediates, and protein synthesis. Viruses are therefore obligate intracellular parasites that rely on entering a suitable living cell to carry out their replication cycle. However, unlike genetic elements such as plasmid, they are also independently present nucleic acid material, but viruses have an extracellular form also. The viral, which is called the viral particle, that enables them to exist outside the host and that facilitates the transmission from one host cell to another. And to multiply, viruses must enter a cell in which they can replicate. The process is called the infection. Now, what are the characteristic features of viral particles or virion? So before we know what are the characteristic feature, we must emphasize why the viruses are called the viral particles, not the viral cell. We all know that there are two types of cells, prokaryote and eukaryote, and the prokaryote cell doesn't have any definitive membrane bound nucleus, but the eukaryotic cell have a membrane bound nucleus, that is a definitive structure that house the genetic material. And so the prokaryotic cell is primitive than that of the eukaryotic cell, as the prokaryotic cell doesn't bear a typical membrane or double membrane bound organelle structure. But still, in case of the prokaryotes, there are some uh, organelle-like structures which are called the inclusion bodies. Those are not typical the organelles that present in the eukaryotes, but they have some compartmentalization within a cell. But viruses are even simpler than that of the prokaryotic ones. They only possess a outside uh, covering and inside a genetic material. We come later onto this. So what are the characteristic features? Although the viruses are not cells, the reason I have already told, and thus are non-living, they nonetheless possess a genome encoding the information they use in order to replicate. However, viruses rely on the host cells to provide the energy materials needed for replication and synthesizing the proteins. Consequently, viruses cannot replicate unless the virus genome has gained entry into a suitable host cell. Virus can exist in either extracellular or intracellular forms. In its extracellular form, a virus is a microscopic particle containing nucleic acid surrounded by a protein coat, and sometimes, depending on the specific virus, other macromolecules. The viral particles or virion is metabolically inert and cannot generate energy or carry out biosynthesis. The virus genome moves from the cell in which it was produced to another cell inside the virion. Once in the new cell, the intracellular state begins and the virus replicates. New copies of the viral genome are produced and the components of the virus coat are synthesized. Viral genomes are usually very small and they encode primarily proteins whose functions viruses can use from their host. Therefore, during replication inside a cell, a virus depends heavily on the host cell structural and metabolic components. The virus redirects host metabolic functions to support the viral replication and assembly of the new variants. Eventually, new viral particles are released and the process can repeat itself. So simply, the viruses consist of a outside covering or a cell and a inside nucleic acid, and that nucleic acid may be of DNA or RNA. And the outside covering is usually a proteinaceous covering which is called the capsid. So this outside covering of the virus is called the capsid. So it's called the capsid, and the capsid is made up of capsomers. So capsomers are nothing but the smaller units which are joined multimerizes with one another to uh, to form the capsid structures. So these smaller units are nothing but the capsomers, and the capsomers are joined with one another to form the capsid. And again, these capsomers are further uh, produced by or made up of some further smaller units which are called the protomers. And the capsomers are the smallest observable three-dimensional morphological units. So the smallest observable unit are capsomers, but capsomers are again made up of protomers. So simply the smaller units, which are called the protomers, join together to form a capsomer, and these capsomers are joined together to form a capsid. And the process of producing the capsid starting from the protomers uh, is called the self-assembly process, because when a single protomer joined with another protomer to form a capsomer, and again when the capsomer joined with another capsomer to form a capsid, this entire process uh, actually uh, leads the driving force to another. So they are self-assembling with one another. They doesn't require any energy from the outside. They produces the one monomer when joined with the another monomer, they produces the energy, and that energy leads to the next reaction. So this type of reaction is called a self-assembly. And the capsomers have specific geometric shapes and symmetry. Hence, the viral particles also have some shapes and symmetry. Usually, there are two major shapes. One is called the rod shaped and spherical shape. So, when the viral particles uh, showing the helical symmetry, they form the rod shaped appearance. So this is a helical symmetry, just like a helix, it moves, and along the helix, these capso capsid molecules are arranged, and it gives rise to a rod shaped appearance. The another symmetry is icosahedral symmetry. This is called the icosahedral symmetry, and when the viral particles show the icosahedral symmetry, they are usually form the spherical shape. And again, in some cases, the in case of particularly the icosahedron type, there are envelope like structure of covering the um, viral particles. So the viral particle may be devoid of the um, any envelopes, which is called the naked viruses, or it may have some envelope like structure, which is called the enveloped viruses. Now, what does the icosahedral symmetry means? Icosahedral symmetry is a special type of symmetry. Uh, so this is a special type of symmetry uh, where a polygon with 12 vertices, that means corners, there are at least 12 corners, 20 facets. Facets means sides. So these are the facets, the triangular shape. These are the facets. So if a polygon contains 12 vertices, 12, 20 facets, and 30 edges, so these white lines are the edges. So when a polygon have 12 vertices, 20 facets, and 30 edges, it becomes a icosahedral symmetrical structure. And the icosahedral symmetry again follow a 5-3-2-fold symmetrical pattern. What does that mean? If we can divide the icosahedral symmetrical structure into these 5, 3, and 2 identical portions. So if this is the icosahedron, and if you look from the upside, so if you look from the upside, so there are 5 planes through which we can divide the icosahedron into 5 identical portions. So this 5 identical portion can be generated when you look from the upside, this point. If you look from this point, so there are 5, 1, 2, 3, 4, 5 identical planes by which we can divide the structure into identical halves. Now if you look or start from the center, just center. So there are three um, planes through which we can divide the structure into identical halves. And another is simply if we cut through this plane, it will form two identical halves. So icosahedron is a polygon which contains 12 vertices, 20 faces, and 30 edges. At the same time, it follows five-fold, three-fold, and two-fold symmetry. Hope you all understand this structure. So this is a human papilloma virus, which shows a round or spherical shape structure. And another, this is a fast virus. We all know the structure of the fast virus, and the head of the fast virus is typically icosahedron. But in case of the fast virus, as this head, this icosahedral head, also associated with a tail and tail pins, end plates, etc. So this is not typically the icosahedron, but along with the icosahedron, there are some associated structure. Hence, this is called the complex symmetry. Now, coming into the nucleic acid structure, as the viruses have two parts: outside capsid and inside nucleic acid. So, nucleic acid, the virus can be classified by the types of the nucleic acid. Like if the DNA is present, it's DNA viruses. If the RNA is present, the RNA viruses. And another is, there is another type where RNA is present, but that is intermediate with a DNA structure, as this type of virus have some reverse transcriptase enzyme which forms a DNA structure from the RNA. So they are also called the retroviruses, and if double-stranded DNA is formed, it's called the hepatnaviruses. And DNA viruses and RNA viruses are further classified into single-stranded DNA virus or double-stranded DNA viruses. So these are some of the types of the viral genome, and along with their host. For example, H1 parvovirus, which is an animal virus and is a single-stranded DNA virus. 5X174 is a bacteriophage and is a single-stranded DNA virus. Similarly, you can find plant viruses also. There's cauliflower mosaic virus is a plant DNA virus, double-stranded plant DNA virus. Cowpea mosaic virus is a RNA virus, but a single-stranded RNA virus. There are similarly some double-stranded RNA virus which infect the plants, animals, and bacteria also. Now the RNA viruses are further classified by the positive and negative strands of the RNA viruses. This is very important. These are the DNA viruses as already discussed, and these are the RNA viruses. And you can see there are some positive and negative signs are within the parentheses. And what does that mean? It's very simple. If you consider the positive RNA as mRNA, that means the positive RNA is nothing but the RNA structure which is capable directly to form proteins, which is capable directly as template for a protein. So negative RNA cannot directly translate it into protein. So whenever a virus possesses a negative strand of RNA, always understand that this negative RNA must have to be converted into the positive RNA, and then it will go for the protein synthesis. And now, how the negative strand of RNA become the positive strand of RNA? By simple replication. Okay, so we all know that if our DNA is replicated, it forms a complementary strand of DNA. Similarly, if a RNA replicated, it forms a complementary strand of RNA, and that complementary RNA is called the positive RNA. So now this is double-stranded RNA virus. So double-stranded RNA virus means there are two strands. So in case of DNA, we all know double-stranded DNA. One strand is act as a sense strand and another strand is act as a anti-sense strand. Similarly, if there are two strands of the RNA, one strand acts as a positive RNA and another strand acts as a negative RNA. So the negative RNA must have to trans replicate it to form the positive RNA, and that positive RNA will go for the protein synthesis. In case of the single-stranded RNA, this is nothing but a positive strand of RNA, so it will directly go for the protein synthesis. And if the single-stranded RNA is a negative strand, then it will first transform into the positive mRNA, positive RNA, and then go for the protein synthesis. And this is a separate class which have a positive single-stranded RNA, but it will go for a double-stranded DNA intermediate by the activity of the reverse transcriptase, and then it will this double-stranded DNA, one of this strand, form the mRNA and go for the protein. So this is the concept of the positive and negative strand. Now you can find that there are some class designated in each DNA and RNA viruses. So these are nothing but the Baltimore classes. We all know that the Baltimore have classified the viruses into 7 distinct classes. So along with the strand structures, the in which class of the Baltimore classification they belongs is also designated. For example, the DNA viruses are include class 1, class 2 and class 7 of the Baltimore classification, and RNA viruses includes class 3, 4, 5 and 6 of the Baltimore classification. And what does this classes means, why they are separated from rest of the viruses have also mentioned here. For example, class 1 DNA viruses class 1, that is classical semi-conservative mode of replication follows, and class 2 a classical semi-conservative replication, but they discard the negative strand of the DNA likewise. So we'll may discuss Baltimore classification in a separate tutorial. Then it will be more clear that why these classes or different classes of bacteria, how these different classes of the bacteria, how these different classes of the viruses have been classified based on their DNA structures. So thank you for today for listening. Please subscribe to the channel.